And it's really truly pleasure for me and a little bit challenge to introduce today's speaker, Dr. Elaine Robey. Elaine Robey is distinguished leader in immunology and a current division head of immunology and molecular medicine in the Department of NCB at UC Berkeley. And the reason why I'm saying it's challenging to introduce her is because Dr. Robey has led a remarkable productive research program for over 30 years, contributing transformative insights at multiple key stages in the evolution of the immunology field. And I will do my best to highlight a few of her many contributions to introduce her research finding and background to all the audience here. Dr. Ruby's research has broadly focused on the molecular signals and the guide T cell development in the thymus and the immune response to Toxoplasma gondii, intracellular protozoan parasite. Her scientific journey began at a pivotal moment just as the first TCR transgenic mice were being analyzed. During her postdoc training, she was the first to propose and test alternative models explaining how TCR recognition of MHC class 1 versus MHC class 2 guides CD8 versus CD4 lineage commitment. Her conceptual framework, contrasting instructive with stochastic or selection models, remains fundamental in our understanding of T cell fate decisions. After establishing her lab at UC Berkeley, Dr. Robey tuned her attention to notch signaling, which at that time was primarily studied in the developmental biology in Drosophila or C. elegans. Her lab was the first to demonstrate that the notch plays a critical role in mammalian immune cell fate decisions, most notably in determining gamma delta versus alpha beta versus gamma delta T cell lineage, pioneering a now well established research direction. Dr. Robey is also a Dr. Robey also applied a new technology to photomicroscopy to the immune system. Her lab early work published in Science provide the first live imaging of Simon-sized stromal cell interactions, revealing their dynamic nature and then laying the foundation for real-time imaging studies in immunology. Her team also led the field in virtualizing T cell dendritic cell interactions within intact lymph nodes, offering crucial insights into antigen presentation and T cell activation. Most recently, Dr. Robey has combined two photomicroscopy with thymic tissue slide technology to dissect the spatial and temporal dynamics of thymic selection. These studies have shed on like key processes such as negative selection, regulatory T cell development, and the TCR tuning during passive selection. Her interdisciplinary expertise using spanning immunology, life cell imaging, computational biology, and microfluidics continues to drive new discovery and provide valuable resources to the scientific community. And today, please welcome and join me in giving warm welcome to Dr. Elaine Robey. Dr. Robey, please.
Thank you so much, Paul. That was a really kind introduction, and thank you all for coming to hear my seminar. So I'm going to be talking today about a project that was very much a team effort. Can you see my pointer when I move it like this?
Yes, I can.
Oh, great. Yeah, so this is very much a team effort between my lab and two other labs, Nir Yosef, who was at UC Berkeley and is now at the Weizmann Institute, and Aaron Streets, who's still here at UC Berkeley. And their student, Zoe Steyer, led much of the work I'm going to talk about. And a lot of people in my lab contributed to this. Dom Yard was a leader in doing experimental testing that I'm going to show. Izzy and Esther contributed also to the experimental testing, and Laura and Lydia helped with the initial experimental design and sample prep. And so the question that we decided to tackle is this one that, as Paul alluded to, it's one that I've been interested in for a long time, even before I started my lab here at Berkeley. And that's the question of how the precursor cell, the double positive precursor cell in the thymus, chooses between the CD4 and CD8 T cell fate. And so, as I'm sure you're all aware, in the periphery, mature T cells respond and recognize pathogen-derived peptides presented by MHC proteins displayed on another cell. And this same recognition of peptide MHC complexes, but engaging with self-peptide MHC, is crucial during thymic development for appropriate cell fate decisions in the thymus. And you're probably also well aware that during thymic development, the antigen receptor gene rearrangements produce a large diversity of different antigen receptors, so that these CD4, CD8 plus double positives, each of them expresses a unique version of the antigen receptor. And in a normal animal, about 95% of these cells will die at the double positive stage, and only about 5% give rise to either mature CD4 or CD8 T cells. And this depends on the low affinity recognition of self-peptide MHC on thymic epithelial cells, whereas high affinity recognition leads to either cell death or death by neglect or agonist selection. And also shown in this diagram is the fact that when thymocytes undergo positive selection on class 1 MHC, they turn into CD8. And when they undergo positive selection on class 2, they turn into CD4 cells. And so the question that we were trying to address, and that I've been interested in for many years, is how is it that the distinct signals generated upon recognition of class 1 or class 2 lead to the establishment of distinct transcriptional networks in T cells? And so this has been really an important model system for understanding mammalian cell fate decisions in general. And it's also very important for the appropriate functioning of the adaptive immune system, because the CD8 cells are cytotoxic cells, and their job is to kill cells that are infected by viruses or tumor cells. And this makes sense because the MHC1 proteins sample endogenous antigens that are made inside the antigen-presenting cell. In contrast, helper T cells, their job is to regulate other immune cells. And so it makes sense that they're associating with class 2 MHC that samples material coming from outside of the cell through phagocytosis. And so this is an important process to keep the cytotoxic responses focused on endogenous antigens and to make sure helper responses are focused on exogenous antigens. And the way that this cell fate decision has traditionally been viewed as if you have a bipotential precursor that symmetrically decides to become either a CD4 or CD8 cell at the same moment in time. But now we know that this view is not correct. And instead, when thymocytes are developing, when these double positives are developing during positive selection, they experience successive windows of opportunity, first for the CD4 fate, and then later for the CD8 fate as the thymocytes are maturing. And we think that this is really important to ensure the appropriate match between the T cell receptor specificity for class 1 or class 2 and the CD4, CD8 fate. And as I'm going to describe in a little bit later in the talk, we're proposing that you have these sequential lineage decisions that also correlate with sequential waves of TCR signaling. And that allows you to have selection for a match, first with CD4 in class 2, and then later with CD8 in class 1. So this is sort of a formal depiction of the idea of a symmetrical fate decision, where as the cells mature, there's a moment in time where their lineage diverges into either the A or the B fate. And this would be a more asymmetric commitment, the type that we think is operating during CD4, CD8 commitment, where the cells, as they mature, first can branch off to the CD4 fate and then later to the CD8 fate. And another thing that's really different about our view of this compared to the way we were thinking about this 20 or 30 years ago is that rather than this being an all or nothing decision, there appears to be a period of plasticity where the cells can, so that the cell fate decision is occurring in kind of a gradual way rather than being a discrete event. And so this provides an opportunity for extracellular signals to impact the ability of cells to adopt one fate or another. And so in terms of this asymmetric commitment model, the idea here would be that all the cells are first auditioning for this first fate, and the extracellular signals are going to determine whether the cells pass or fail the audition. And we've known for a little while now about these key transcription factors that control the CD4 versus CD8 decision, where TH-PUC is a transcription factor that specifies the CD4 lineage, and RUNX3 is a transcription factor that specifies the CD8 fate. And we also know that these two transcription factors mutually repress each other's expression, and that TH-PUC has a positive autoregulation loop that helps to lock in the CD4 fate. And it's also been appreciated for some time that the recognition between class 1 and class 2 differs quantitatively, in that class 2 recognition seems to result in stronger or more continuous signals, whereas class 1 recognition results in weaker or more intermittent signals. But how these differences in TCR signaling link up and specify these different transcriptional programs has really remained largely a black box. So we reasoned that the emerging techniques of multiomics could provide an opportunity to make a high-resolution gene expression map as CD4 and CD8 cells are maturing and as their lineages diverge. And this kind of temporal map of gene expression would be very helpful for developing a full mechanistic understanding of the lineage choice. So why is this important? Well, for one thing, it helps us to pinpoint the lineage branch points, so we can now start to see what is the earliest developmental window where the gene expression changes start to diverge. And that allows us to do differential tests to identify candidate drivers of the lineage differences. And I'll show you how we did that. Also, there are multiple branches downstream of the T cell receptor signaling pathway is indicated here. And these branches of downstream the signaling pathway culminate in different activation of different transcription factors. And so we were hoping to uncover whether different branches of the TCR signaling pathway are selectively activated and required in one lineage or the other, based on the temporal and lineage-specific expression patterns of these TCR target genes. So the challenge to generating this high-resolution map is in part the fact that this is taking place in this complex three-dimensional architecture of the thymus, and it's a lengthy process and involves contact with all these support cells. And at any moment in time, if you look in the thymus of a young animal, you'll find thymocytes representing all the different stages of development. And these thymic progenitors are extremely heterogeneous, both because of their developmental stages and also because of the different T cell receptors that they express. So to deal with the heterogeneity from the T cell receptor expression, we used a very common trick that mouse immunologists love to do, which is to use mice that are engineered to express a single rearranged T cell receptor alpha and beta gene. And what this does is that now, instead of having a very diverse set of thymocytes where it's very difficult to infer the specificity of these thymocytes for class 1 or class 2, if we use a mouse, and in this example, this is a mouse that expresses a T cell receptor transgene that is specific for class 1 MHC, in these animals, all of these thymocytes essentially are going to become CD8 T cells. And so that helps us to address the heterogeneity in the T cell receptor by using these kind of models. In terms of addressing the heterogeneity of the developmental stages, there we turn to multiomics. And I wanted to first just describe the traditional way that we've tried to look at heterogeneity within the thymus is to use flow cytometry. And here's just a typical flow cytometry plot looking at CD4 versus CD8 expression, where the majority of cells in the thymus co-express CD4 and CD8. These are the precursors that are undergoing positive selection. And then at the end of the process, you have CD4 and CD8 single positive thymocytes. And then people have used a variety of different markers to try to subdivide these populations based either on additional markers and in some cases on the level of CD4 and CD8. However, this approach is very subjective. The way that people draw the gates differs from lab to lab. And also, in many cases, the developmental order is ambiguous. So we don't really know which population is coming before the other. And this type of fractionation also always results in contamination. So you have cells that are undergoing negative selection or becoming Tregs or unconventional T cells that are mixed in with these cells that we think are the precursors and the endpoints to the process. So multiomics lets you get around this process. And we used a method called site-seq, which is basically like single-cell RNA-seq. But in addition to measuring the transcriptome of the individual cells, we're also measuring their cell surface protein expression using antibodies to cell surface proteins that are labeled with DNA barcodes. And so we can simultaneously measure cell surface protein expression and the transcriptome on individual cells. Now, the thymocytes we're using here are coming from either wild-type mice or mice that are restricted for a particular lineage. And I alluded already that if to these T cell receptor transgenic models, so OT2 and AND are two different transgenic models that express class 2 specific TCRs that restrict the development of these cells only to the CD4 lineage. And we also used mice that are deficient for class 1 MHC. So in these mice, the thymocytes are, they're diverse, they have diverse receptors, but only cells that are specific for class 2 can undergo positive selection since class 1 is missing. For the CD8 lineage, we use the same approach where we have two different class 1 specific T cell receptor transgenes that lead to positive selection in the CD8 lineage. And we also use class 2 deficient mice that have diverse receptors, but positive selection can only happen on class 1. And then so we generated this large data set, and then the Joseph lab, near Joseph's lab, had developed this very useful set of tools called Total VI, which allows for joint probabilistic modeling of the transcriptome and the protein measurements on individual cells. And so when this data was analyzed and looking at the initial clustering, we were able to resolve many different populations of thymocytes based on their similarity in gene expression. And so I just want to point out here that these populations, which represent double positive thymocytes that are receiving T cell receptor signals, as well as CD4 cells that are just recently positively selected and the more mature, and the CD8s that are at various stages of maturity. So we can identify these from this data set. And there are many other interesting populations here, which we and others have been exploring. But for now, we're going to exclude these populations and just focus on the cells that are undergoing positive selection. And we've provided this data set as a resource in a very user-friendly format. And it's been, I hope, a helpful resource to the community of people working on thymus biology. So now if we pull out those cells that are undergoing positive selection and we recluster them, the UMAP looks like this, where CD4 cells and 8 cells are separating out here on the right part of the UMAP. And on the left, the double positives are intermingled. And the class 2 specific CD4 faded cells are shown in different shades of red. And the class 1 or CD8 faded cells are shown in different shades of blue. And we think this is a very unique, there certainly have been other studies published using single-cell RNA-seq approaches to look at thymic development. But our data set is very unique because we have this dense sampling of the intermediate states. And also we define the cell state both by the transcriptome and by the cell surface proteins, which provides a more accurate definition of cell state. And also because we're using these genetic models that are lineage restricted, we can infer the differentiation, the commitment of the cells before their overt differentiation. So one of the analysis that we've done here is to use a technique called pseudotime analysis, which is basically a similarity-based trajectory inference, where every cell in this positive selecting data set is assigned a pseudotime or a position on the trajectory going from early double positives to the later CD4 and CD8 single positives. And we feel that this method is very robust. And it was very reassuring to see that when you look at gene expression, so here we're just looking at the relative gene expression of two different mRNAs that we looked at and two different proteins based, these are genes that are, or proteins that are known to be either up or down regulated during positive selection as thymocytes mature. And red is showing the average data for the CD4 fated cells, and blue shows the average data for the CD8 fated cells. And you can see that the pseudotime trajectories of the CD4 and CD8 fated cells are very well aligned. And this is because the majority of gene expression are correlated with the development of the cells, not their lineage choice. So this provides a very robust relative developmental order for the data for the cells in the data set. So that was for genes that are similar between CD4 and CD8, which are the majority. But when we focus in on those genes that we expect to be differentially expressed between CD4 and CD8, we see some interesting patterns. So here now we're looking at the average expression of individual genes over pseudotime. This is looking only at CD4 fated cells, so cells which are class 2 specific. And in lilac here is shown expression of GATA3. GATA3 is a transcription factor that is an upstream regulator of Th-Puc, and RUNX3 is that CD8 associated transcription factor I mentioned before, and that's shown in green. So we, as expected, we saw that the GATA3 expression comes up during the earliest phase of positive selection, and it's followed by Th-Puc at a later pseudotime, and RUNX3 remains low. And that's the expected pattern for the CD4 fated cells. But when we looked at the CD8 fated cells, we were surprised to see that in the early parts of the trajectory, they look very similar to CD4 fated cells. In other words, there's an induction of GATA3 followed by an induction of Th-Puc at the same pseudotime values. And so we started calling this period, this early phase of positive selection, a CD4 audition because of this parallel pattern of gene expression that appeared as though the cells were trying out the CD4 lineage. Later on, and the cells were at a more mature stage, the CD8 fated cells then upregulate RUNX3, and this corresponds to the downregulation of Th-Puc at a stage where Th-Puc remains high in the CD4 cells. Now, it's interesting to compare this pattern to what happens with T cell receptor signaling. And here we're looking at expression. This is a similar kind of plot where we're looking at average expression versus pseudotime for CD4 fated cells and CD8 fated cells. But now we're looking at two well-characterized T cell receptor target genes, EGR1 and CD69. And what we found was that at this earliest phase of positive selection, the CD4 fated cells have a more prolonged early wave of TCR signaling, and this overlaps with the period of the CD4 audition. And this makes sense because of this data I described earlier, suggesting that class 2 leads to stronger or more prolonged signals. But what was more surprising to us was that if you look later in pseudotime in the CD8 lineage, you see this distinct wave of T cell receptor signaling. And this was particularly intriguing to us because the second wave, this late wave of TCR signaling, overlapped in pseudotime with the induction of RUNX3, so corresponding to the specification of the CD8 fate. And so putting this all together, we can come up with this kind of temporal map where at the very first stage of positive selection, all thymocytes first audition for the CD4 fate during an initial wave of TCR signaling. And then for the CD4 fated cells, they experience this prolonged wave of signaling that leads to full TH-puck induction in the CD4 fated cells. On the other hand, for CD8 fated cells, that initial TCR signaling is more transient, leading to a failed CD4 audition. And then at a later stage of development, the cells then acquire the ability to upregulate TCR signaling again, and now they can undergo CD8 lineage commitment and upregulate RUNX3. Now, as I mentioned, the pseudotime provides a relative developmental ordering of these events, but it does not give us an absolute measure of time because it's really just based on the changes in the gene expression. However, there's historical data that can provide landmarks for some of these events. For example, it's known that from the time you initiate positive selection, it takes 1 or 2 days for cells to appear as mature CD4 cells, whereas it takes 3 to 10 days or more for CD8 cells to appear. So this gives us an idea of how long these events actually take, and also show that these gene expression changes are taking place over a prolonged period of time. And so based on all this data, we've proposed what we call a sequential selection model that helps to explain the link between MHC recognition and CD4-CD8 fate. And we described this model in a couple of reviews that we published recently, but I just want to briefly take you through this model. So this is addressing the question of how does the recognition of MHC direct the lineage choice? And another related question you could ask is, why do CD4 and CD8 T cells develop sequentially instead of simultaneously? And we would argue that this is important because by having these sequential developmental events, it's possible to have sequential selection to ensure that the TCR signal occurs because you have a match between CD4 or CD8 expression and the specificity of the T cell receptor for MHC. And actually, this is very reminiscent of the stochastic selection model, which my colleagues and I had proposed way back when I was still a postdoc. And then the next question is, why do CD4 T cells develop first? And I think here it's interesting to note that CD4 cells in general, they seem to have intrinsically stronger T cell receptor signals. And this is an image from one of Chris Hoquist's papers using a GFP reporter for TCR signal strength. And what you can appreciate here is that both in the thymus and in the periphery, the CD4 cells are experiencing stronger TCR signals than the CD8 T cells are. So the model that we propose takes all of this into account, and it incorporates elements of earlier models, including stochastic selection instruction, and also the kinetic signaling model of Al Singer. And so here's how that works. So this is the same scheme I showed you before, but now I've just divided them up. So we have CD4 fated, what's happening to the CD4 fated cells on top, and what's happening to the CD8 fated cells on the bottom. And so the first step is the CD4 audition. And at this stage, we would propose that this has to happen in a limited time, that from the time positive selection starts, you have this limited time window to get enough signal to become a CD4 cell and to fully upregulate TH-puck. So it makes sense that the CD4 cells, with their stronger overall signal, are going to be more efficient at completing this process. And that's going to favor the CD4 fate as that first commitment opportunity for this positively selecting thymocytes. Now, for those cells that when this process occurs, there's a downregulation of CD8, which is not a problem for these cells because they're recognizing class 2. But the cells that are fated to become CD8, their receptor is specific for class 1, So when they start to downregulate CD8, they lose the signal, and this serves as a check to make sure that thymocytes that are specific for class 1 fail the addition and don't become CD4s. And I just put this cartoon in here just to remind you that CD4 and this TCR that's specific for class 2 have to both engage for this cell to undergo positive selection. Now, during the second wave, the CD8 has come back up, and now the cell has a chance to check to see if the co-receptor, in this case CD8, matches with the T cell receptor. And so this is another selection step to ensure that if you had class 2 specific cells, that maybe they just got a weaker signal and they weren't able to complete the CD4 addition, they're going to be eliminated here so that you don't get mismatched cells that have class 2 specific receptors adopting the CD4 fate. So we think this model incorporates really basically all the data that's out there and gives us a way to understand how you can have this very strict linkage between the MHC specificity of the T cell receptor and the expression of CD4 and CD8. Let's see how I'm doing on time. Okay, it's been 31 minutes, so I'm okay. Okay, so I wanted to talk about another aspect of this data that we found really interesting, and that's asking the question about how do these TCR target genes change during positive selection? And can we infer from that about how the T cell receptor branches of the TCR signaling pathway are changing? And so for this part of the analysis, Zoe performed differential expression tests to identify these TCR transcriptional changes that differ between the two lineages. And so this is how this works. So she's basically comparing CD4 and CD8 fated cells at bins across the pseudotime. And you can see that in the beginning of the pseudotime, there are very few gene expression differences. But as you progress along the pseudotime axis, you accumulate more and more gene expression differences. And it turns out there's about 300 genes that are significantly higher in CD4 compared to CD8 fated cells in at least one pseudotime bin. And so we call these CD4-DE genes. Now, many of those are TCR target genes, and we've analyzed this in a variety of different ways, and all 3 different ways, independent types of analysis, that all point to a role for NFAT downstream of the TCR signaling pathway at this earliest phase. And this is just that cartoon I showed you before of the TCR signaling pathway, where I've highlighted NFAT, which is downstream of calcium and calcineurin in this pathway. So I want to describe a little bit more about how this analysis worked. So I mentioned there are 300 genes or so that are higher in CD4 than CD8 at some point along the pseudotime. And so to organize these genes into categories, Zoe performed this cluster analysis. So each of these clusters now represents a temporal or lineage-specific expression pattern of the group of genes. And I want to highlight here these 2 clusters, cluster 4 and cluster 7. And the reason that these clusters caught our attention is for 2 reasons. One, these clusters contain the earliest differences. Like, so for example, cluster 1, these are clearly higher in the CD4 lineage later than CD8. Here's another example, cluster 5. But these are genes that are important after lineage commitment. They're more markers for CD4 fate. So these clusters 7 and 4 had the earliest differences showing up. And also, these 2 clusters were the ones that were significantly enriched for T cell receptor target genes. So we dug in more to these clusters, and they're shown here. And it was interesting to us also that this cluster 4 had contained some known regulators of TH-PUC, including GATA3 and also LEF1. Both of these are known to be upstream regulators of TH-PUC. So we asked computationally, what are the transcription factor activities that drive this expression in this earliest, the TCR target gene expression in this earliest wave? of TCR signaling. And so we did this using a method called CHEA3 analysis, which is a tool that takes a set of target genes and ranks transcription factor activities that are most likely to account for that activity. And so I'm showing you here these two clusters and the ranking score from this tool. And I've color-coded these based on the branches of the TCR signaling pathway. And so what you can see here is that NFAT shows up here in this cluster 4, the one that's earliest and that does not have a second wave in the CD8 lineage. Whereas the this cluster 7, which has both a first and a second wave, are expressed, genes are expressed throughout positive selection. So it looks like the MEK-ERK targets are more general and the NFAT targets appear to be more specific for that early window. So what is the role of calcineurin NFAT in positive selection and lineage commitment? There was already a lot of data for a role for MEK-ERK, but the role of calcineurin NFAT has really remained somewhat mysterious. So there were early experiments where mice were treated with the immunosuppressive drug cyclosporin A, which is an inhibitor of the calcineurin NFAT pathway, and that does lead to a block in T cell development. However, when the people looked at this more closely, it turned out that the role of NFAT that was being revealed from this long-term blockade actually was prior to positive selection. It appeared to be a defect with beta selection that was then reading out during positive selection. And so because of that, the role of calcineurin NFAT downstream of TCR signaling during positive selection had remained unknown. So we tackled this using our favorite experimental system, which is a thymic slice culture system. And so in this example, we prepared vibrotome cut thymic slices from newborn mice, and we cultured them in vitro for several days, and then read out the development by flow cytometry. And we observed that there's a wave of CD4 and CD8 single positive development that occurs during the neonatal period that you can capture using this neonatal culture system. So then Dom Alliard went in and looked at this development in these slice culture and asked what effect the calcineurin inhibitors would have on development in these cultures. Now, the way he's done this analysis is using 7 cell surface proteins and 3 lineage-defining transcription factors, and he used multidimensional computational gating to analyze the data. So if there's no drug, you culture the neonatal slices for 3 or 4 days. This is what you see, where it goes from double positive to CD4 single positive here and CD8 single positive here. And orange here are transitional cells that are transitioning between the double positive and the CD4 single positive stage. In the presence of the calcineurin inhibitors, there's a selective loss of this intermediate population. Whereas if you use very minimal concentrations of MEK inhibitors, this ablates both CD4 and CD8 as well as these intermediate cells. So this suggested that this could fit with the idea that calcineurin was playing a more selective role early in positive selection. Now, our model also predicted that calcineurin should repress GATA3 expression in these double positive thymocytes. And indeed, that's what we found when we looked, if we gate now on the thymocytes that are double positives and that are showing signs of TCR signaling, we see that these cells now have lower expression of GATA3. We see this both at the signaled double positive stage as well as the CD4 positive CD8 low stage. So both the bioinformatics and the experimental testing support a role for calcineurin NFAT downstream of TCR in this early induction of the CD4 fate during positive selection. So I think I'm just ready on time here. So to just wrap up, I'll just mention where we're heading with this now. We're interested in understanding more about the maturation-associated changes that set the stage for these two different signaling waves, as well as these two different cell fate decisions. And we're particularly interested in changes in how the T cells receive TCR signals during this process. We're also interested in understanding more about the molecular pathways for TCR signaling for the TCD4 addition, and we have some other candidates from the bioinformatics that we're now trying to test. And finally, the issue about this CD8 specification still remains quite unclear, and we're trying to use this similar kind of approach to understand in more molecular detail how this works. And so again, I'll just acknowledge the amazing team of scientists that I've had the privilege to work with, and I'd be very happy to take any questions.
Wow, so interesting, and